Hello, everyone. Thank you all for attending today's virtual workshop. And depending on where you are joining us from, good morning, good afternoon, or good evening. And welcome to today's session of IPRP Biosimilar Working Group Scientific Workshop on increasing the efficiency of biosimilar development program and reevaluating the need for the comparative efficacy studies. And today's session is the first public session that we are going to hear from the regulator's perspective on the need for comparative efficacy studies in a biosimilar development program. And I'm Carol Kim. I'm a scientific reviewer in the Office of Therapeutic Biologics and Biosimilars within the US FDA, closely working with Dr. Sarah Yim, who is the chair of the IPRP Biosimilar Working Group. And I would like to first thank all members in IPRP Biosimilar Working Group, as well as the subcommittee Biosimilar Working Group members for attending many planning meetings and contributing their valuable time and sharing their expertise in effort to make this workshop very successful. And with that, I am excited to get started with today's agenda. Next, please. As a few housekeeping remarks, please note that as an attendee, you will not be able to use your microphone to speak during the seminar or during the webinar. That means the chat, the microphone, and video are disabled for the attendees. And when the time comes for the attendee question, please use the Q&A function to provide any question that you would like to have it addressed by our panelists. And finally, please note that today's session is being recorded, with the recording to be made available on the IPRP website in the coming weeks, along with the presentation material and speaker biographies. Next, please. Here's an overview of today's session. Following my brief introduction of what this workshop is about and what we hope to achieve in this workshop, in today's public session, we will have the presentation and panel discussion by US and international regulatory expert representatives in the area of biosimilar development. And they will be sharing their experience accrued with comparative clinical efficacy studies up to date. And Dr. Ali Al-Humaidan from Saudi Food and Drug Authority will be moderating the panel discussion today. And then I'll provide a brief closing remark and we'll end today's session. Next, please. For those of you who may not be familiar with who we are, I would like to briefly talk about IPRP Biosimilar Working Group. The IPRP Biosimilar Working Group was established to discuss emerging regulatory issues related to the biosimilar product. And Dr. Sarah Yim from US FDA is the chair, and Dr. Ali Al-Humaidan from SFDA is co-chair, leading this IPRP Biosimilar Working Group. And the main scope is to discuss the regulatory challenges and potential topics or the areas for harmonization regarding the biosimilars. And we hope to consider how the regulatory convergence can be achieved by sharing and exchanging the information without compromising the confidentiality. And we also hope to explore the work sharing process with other international bodies and collaborate in terms of a training of international regulators. And we have two main objectives. And the first one is for the regulatory conversion of, to have a regulatory conversion of a technical requirement for the biosimilar product in facilitating the regulatory process. And the second objective is to provide a support to the international regulators so they can develop safe and effective regulatory framework for the biosimilar products. And our members are involved globally. and they are listed under this participant in this slide. Next, please. From a US FDA perspective, in our effort to streamline the biosimilar development program, we hosted 2 workshops in the past. And in September 2021, in collaboration with the Duke Margalit, we hosted a virtual workshop entitled Pharmacodynamic Biomarkers for Biosimilar Development and Approval. And in this workshop, we had discussions on how to streamline and improve the efficiency in biosimilar product development programs, and focused our discussion on how we can leverage the PD biomarkers in place of comparative efficacy studies. And in September of 2022, we hosted a virtual workshop entitled Increasing the Efficiency of a Biosimilar Development Program. And in this workshop, we focused on how we can leverage the comparative efficacy study information if we decide to use that. And we had some detailed discussion on innovative statistical methods. Next, please. So the purpose of this year's IPRP workshop is to focus our discussion more on how we can reevaluate the need for the comparative efficacy studies to increase the efficiency in a biosimilar development program. And we divided this session into 2 parts. The first one is a public session, and the second one is a regulator session. So for today, for the first day of a public session, you will be hearing from the regulator's perspective on how they've been using this comparative clinical efficacy study in a biosimilar development program. And tomorrow, you will be hearing from a stakeholder's perspective on the pros and cons of comparative efficacy studies being used in a biosimilar development program. And next week, we will have 3 days of a regulator session, and we will continue to have a discussion on the regulatory consideration for streamlining the biosimilar development program. And we also hope to have more discussion considering around the comparative efficacy studies when those studies may or may not be needed. Next, please. So with that, I would like to introduce today's moderator, who is Dr. Ali Al-Homaidan. He is a highly accomplished executive with a track record in the pharmaceutical and biotech sectors. And he's currently serving as the vice chair of the IPRP Biosimilar Working Group. And he has devoted over 2 decades to Saudi FDA. And notably, he served as the executive director of product evaluation and standard setting, as well as the director of a biological product scientific evaluation. And his academic credentials include a doctorate in biotechnology from the University of Queensland, Australia, and received a certificate of management excellence from Harvard Business School. And his extensive experience combined with his academic achievement underscores his commitment to advancing the pharmaceutical and biotech industries. And with that, I would like to turn it over to Dr. Ali Homaidan. Thank you.
Thank you, Carol. Hello, everyone, and good morning. Next slide, please. Our first speaker of the day is Dr. Marie-Christine Bilski, MD from the MHRA, United Kingdom. After studying medicine in Strasbourg, France, she made a career in pharmaceutical industry, where she participated in the clinical development of a wide range of medicines, including orphan and biological drugs. In 2006, she joined as a medical assessor for the Biologics and Biotechnology Unit of the Licensing Division at the United Kingdom Regulatory Agency. She was a member of the Biosimilar Medicinal Products Working Party at the European Medicines Agency, where she participated in the drafting of several biosimilar guidelines and in the assessment of several biosimilar products, including insulin, infliximab, rituximab, and bevacizumab, and adalimumab. She was part of the drafting group of the guidance on the licensing of biosimilar products issued by the MHRA in 2021. Dr. Marie-Christine, please take it away.
Good morning, good afternoon, good evening. I will shoot immediately with the UK MHRA guidance issued in May 21 on the licensing of biosimilar products. So this was only possible after the UK had left the European Union, as you may well know, in January 2020. Before that, the legal framework for applications of biological products manufactured using recombinant technology was obligatorily the centralized procedure, which means an assessment by the EMEA, the European Medicines Agency at the European level. And being part of this network, the MHRA was actively participating in the assessment of biosimilars, and this allowed us to develop our streamlined approach to the development of biosimilars. Actually, based on this experience, over 8 years of, we reviewed the applications and the evaluation of 20 biosimilars to 5 monoclonal antibodies and 1 fusion protein. And in this review, we deliberately focused on these complex molecules for which there is no pharmacodynamic marker, because already at that stage, the use of pharmacodynamic studies instead of efficacy studies using surrogate markers, this use was already considered acceptable by most regulators. So this was not our approach. We wanted to see how clinical trials could contribute to the assessment of complex molecules such as monoclonal antibodies. And our review did not identify any instance where efficacy trials added crucial information to establish biosimilarity. Moreover, there were a few studies where efficacy or immunogenicity findings showed some differences, but these were finally discarded based on the remaining evidence, meaning the analytical testing and the clinical PK information. And we were not surprised by these results, because we consider that efficacy trials are the least sensitive tools of the comparability exercise. They're clearly less sensitive than analytical tests and are even less sensitive than PK or PD data. Therefore, we think that no efficacy trial would be able to resolve analytical uncertainties, and no clinical trial could be used to rescue a failed comparability exercise on the level of the quality. And that's why we considered most efficacy trials are not an efficient use of limited resources, in particular of patients who should be enrolled in studies on innovative medicines rather than in biosimilarity trials. And this review was published in 2020, and it enabled us to conclude that in-depth knowledge of the reference product with high-performing analytical tools largely predicts clinical comparability subjects to confirmation by a comparative pharmacokinetic trial. So our guidance is the following. First of all, I want to emphasize that it does not systematically waive clinical trials. We require the applicant to justify that efficacy, safety, and immunogenicity of the specific biosimilar they have developed can be derived from the knowledge of the reference product and, very critically, its mechanism of action. So what we expect in the application is a justification that comparable efficacy of the biosimilar and reference product can be derived from comparable binding properties, functional characteristics in relation to the mechanism of action of the reference product. And likewise for immunogenicity and safety, the justification for comparability is informed by clinical experience and quality attributes of the reference product. For example, drug product characteristics like protein aggregates, impurities, formulation of the biosimilar candidate, all these should form the basis for justification that the safety and immunogenicity are comparable to those of the reference product. And this argument finally is supported by the data from the confirmatory PK trial, which should show comparable safety and immunogenicity. We added two statements in the biosimilar guidance. First of all, the fact that if a biosimilar candidate has been shown to be highly similar to the reference product in terms of analytical characteristics and functional properties related to the mechanism of action, all the indications of the reference product are granted without further justification. And last year, we updated this guidance with a statement on interchangeability, which is in line with the EMEA approach, meaning that once authorized as a biosimilar, we consider that biosimilars are interchangeable with reference product and amongst themselves. So in practice, what impact has had our guidance? First of all, unfortunately, it came out in the middle of the pandemic. So the period was not extremely active in terms of new biosimilar applications. However, we have been holding a lot of scientific advice meetings where we have discussed with companies biosimilar development without the need for an efficacy trial. The problem we see in practice is that most companies are obliged to conduct efficacy trials regardless, because of requirements from other jurisdictions, especially the FDA and EMEA. So what can happen is that we receive applications earlier before the completion of the efficacy trial and the submission to these other jurisdictions. This is not very satisfactory in our mind, to say the least, and we have great expectancies about this meeting. We do hope that it will open a path to greater alignment of requirements across regulatory agencies, so that patients will finally be invited to test innovative medicines rather than biosimilars. Thank you for your attention.
Thank you, Dr. Marie-Christine. Okay, if there are any questions, please feel free to type them in the questions and answers, and we'll highlight them after the presentations. Next slide, please. Our next speaker is Dr. Hina Kang, PMD from the WHO. Dr. Hina Kang is a scientist in the Norms and Standards for Biological Products team at the WHO, Switzerland. Dr. Kang joined WHO headquarters in January 2009 and has been in charge of the development and implementation of the WHO guidelines for regulatory evaluation, of biologicals, in particular biotherapeutics, including biosimilars. She has coordinated the works to provide regulatory principles in biotherapeutics area and organized many workshops. She has also coordinated works to develop case studies and published many articles to implement the evaluation principles of the WHO guidelines into regulatory practices in several countries. She is a member of the Biosimilar Working Group of the International Pharmaceutical Regulators Program. Prior to joining the WHO, Dr. Kang was a scientific officer for 12 years at the Korean Ministry of Food and Drug Safety, who was responsible for reviewing license applications as well as quality control tests and facility inspection of bacterial vaccines. In 2004, she worked on the project to develop HCV DNA vaccines at the Vaccine and Infectious Disease Organization International Vaccine Center in the University of Saskatchewan, Canada. Dr. Hina, please take it away.
Thank you. Marcos, could you put my slide on the screen, please? Thank you. First of all, thank you for giving me a chance to share the WHO activities related to regulatory standardization of biosimilars. Next slide, please. Today, I will cover the implementation of the resolution in 2014 and outcome of review and survey conducted in 2019 and 2020, and finally, the key updates made in the revised guideline in 2022. Next slide, please. The first WHO guideline on evaluation of biosimilar issued in 2009, and in 2014, WHO Health Assembly resolution was adopted. The resolution was the first ever resolution in biotherapeutic area in WHO. So it's a really meaningful milestone to work in the area of biosimilar. And one of the requests from resolution was to convince expert committee on biological standardization to update the 2009 guideline. But there are two points should be considered for the revision. The first one is taking into account the technological advances for the characterization of biotherapeutics. And the second point was considering national regulatory needs and capacity. So to implement in the resolution, WHO has organized a lot of activities. For example, we coordinate the review of current scientific evidence and experience gained in the last decade. And also we have organized many meetings, including the informal consultation in 2021. And also we coordinate the survey in 2019 and 2020. Next slide, please. Yeah, I would like to share some outcome from the review conducted in 2020. The aim of review was to review the scientific evidence and experience to identify issues, cases for further reducing non-clinical and clinical data, to reach consensus on regulatory consideration and expectation for evaluation of biosimilars, and to update the guideline, 2000 guideline, with providing more flexibility. For the methodology, the review of the relevant guideline, including US FDA, EMA, and Health Canada, was conducted, and also we have a publicly available literature publication were reviewed. It includes EPA from EU, and also a lot of journal publication, particularly for long-term efficacy and safety of biosimilar, particularly for the year in 2017 to 2020, and also the review included systematic review publications to cover some old data. And finally, the review try to evaluate the roles and relevance of clinical efficacy studies for the benefit risk assessment of biosimilars for the possibility to reduce the clinical data requirement. And I would like to highlight some key findings from the review. And the long-term safety, efficacy, and immunogenicity data of licensed biosimilars since 2006 do not raise any concerns, and current data could suggest the state-of-the-art analytical and functional testing and robust PK and PD studies are sufficient to demonstrate biosimilarity, where in vivo animal studies and large confirmative efficacy and safety studies are generally not needed. So the WHO 2009 guidelines should be updated to reflect the current scientific knowledge. Next slide, please. We also conducted a survey in 2019 and 2020. The aim of survey of 2019 was to describe the progress made and the regulatory landscape changed for biosimilar in 21 countries during the last decade. After the issue, the first WHO guideline in 2009, we conducted a survey in 2010. So the survey outcome from 2019 and 2020, the results were compared with the outcome from 2010 survey. And the aim of survey in 2020 was to identify challenges and areas where further support to member states needs to be provided. The survey in 2019 actually focused on situation regarding the regulation and guideline terminology issues and approval or development of biosimilars, and the outcome from 2019 survey were incorporated into the revision. And the survey in 2020 was focusing on the challenges related to reference product resource, like a lack of resource and quality of biosimilars, and issues related to the use, for example, labeling, naming, or interchangeability issues. The outcome of 2020 survey, actually, the purpose was to plan the WHO activities after the revision of the guidelines. Next slide, please. So the review and survey outcome have been published in open access journals. So feel free to read if you want to have more details. Next one. Next slide, please. Yes. Yeah, and I would like to highlight some key updates incorporated into the revised guideline issued in April last year. Actually, we changed the terminology from similar biotherapeutic product to biosimilar because similar biotherapeutic products, as it says, it only covered the therapeutic product, but the 2022 guideline, the revised one, covered all biological product. That's why we changed the name and the terminology. Next one. And scope of guideline also changed it. As I mentioned before, it covers entire part of biological products. And also some of the principle of guideline could apply to the low molecular weight heparin and recombinant analog of plasma-derived products. But vaccine and plasma, the plasma-derived products are excluded. So only the recombinant analog of plasma-derived products are covered. Just for your information, there was some discussion on the vaccines. So far, vaccines were excluded, but in near future, maybe the vaccine, particularly the mRNA vaccine, can be considered because it can be characterized well. Next one, please. Also, we deleted the concept of a stepwise approach because from the experience, we understood stepwise approach is not relevant because more like a concurrent fashion approach is used in the practice. Next one. The clinical evaluation, there are really meaningful changes made in clinical evaluation part. For example, in 2009 guideline said the similar efficacy of a biosimilar and choose reference product will usually have to be demonstrated. And also it said in certain cases, comparability PK, PD studies may be approached, but in 2022 guideline, the other way around. We said a comparative efficacy and safety trial will not be necessary. And prior, we said that the starting PK and PD comparability isn't generally required for clinical evaluation. So the nuance is a little bit different. And also we tried to provide more flexibility for the efficacy trial. Next one. Yeah, and also the immunogenicity studies in 2009 guideline, the immunogenicity data should always be provided, but in 2022 guideline, immunogenicity study may not be necessary. Yeah, so it's try to provide more flexibility and less weight on clinical parts of the evaluation. Next one. Yeah, thank you for your attention, and thank you again for WHO drafting group.
Thank you, Dr. Hina Khan. Next slide, please, Carol.
Hi, Marcus, can you advance the slide? We may be having some IT issue here. Marcus, can you advance the slide?
Hey, Carol,
Because-
you're mute.
Oh, all right. Can you hear me now?
Yes, I can hear you.
Okay, yeah. We need to have next slide present.
You are still mute.
No, you aren't, Carol.
Okay.
Yeah, Marcus. Okay, let me type. Can you advance the slide, Marcus?
I don't know whether you can hear me, Marcus. Could you advance the slides? That's what Carol has been saying.
Yeah, thank you. Great. Okay, now it's coming. Thank you.
There we go. Thank you, Marcus. So our next speaker is Dr. René Anwar, MD from the European Medicine Agency, European Union. Dr. René is a senior clinical expert at the Austrian Medicines and Medical Devices Agency, where he coordinates scientific advances with industry and serves as lead clinical expert in numerous European marketing authorizations. He is furthermore involved in the European Medicine Agency's international cooperation platform. He has more than 10 years experience in the biosimilars area, becoming a member in the European Medicine Agency Biosimilar Medicinal Product Working Party in 2020, and recently being elected chairman of this group in July 2023. He is a member of the HMA Biosimilar Working Group, an initiative to increase the uptake of biosimilars in the European countries. Dr. René, please take it away.
Yeah, next slide, please. Thank you for the introduction, and good morning, good afternoon, and good evening also from my side. The first part of my presentation might not really come as a surprise to most of you, because the European position on that hasn't changed that dramatically in the last years. So next slide, please. What is our general philosophy? When do we require clinical efficacy trials in the biosimilar development? Why do we require them? And the aim is really to address slight differences that we have encountered in previous steps. And this is really the key point: confirmation that slight differences that we have seen do not matter as previously assessed on quality or PK level. The clinical trial should not serve to really account for larger problems encountered on earlier levels, so it cannot correct them. And this is the philosophy. The truth is that we have come a bit in the day-to-day business concerning the assessment of those products into the direction that more than we see this as a pyramid, we are talking about the totality of data. So it happens also, one could argue a bit against that philosophy sometimes, that we are going back and forth, but the result is, and I believe that is not a bad way to approach this actually, that we take the whole development into account and see how problems translate from one level to the other, and get quite a comprehensive picture about biosimilarity. Next slide, please. So when are efficacy trials necessary? So usually they are, but actually we are at the point where in most cases the product falls into one of many exceptions that do not require one. I'll be a bit more specific about that in a minute. How should they be designed? They should be adequately powered, randomized parallel group comparative clinical trials, preferably double-blind and using efficacy endpoints. The study population should be representative of approved therapeutic indications, and this is perhaps more important, sensitive for detecting potential differences. In general, we prefer an equivalence design, inferiority approaches, and if there is a small sleeping beauty hidden in what we require, that might be it. We do not see that this is used too much or argued for too much too often in scientific advices. I believe developers could focus more into that direction and also discuss with their authority why they think that a non-inferiority design might be applicable for their product. Next slide, please. Okay. I was talking about exceptions just a second ago, and we do have a lot of product-specific guidances for biosimilars, and those actually have already stated that for most of those actually PK/PD approaches are relevant and no clinical trial is needed anymore. sort of an exception to that are the biosimilar monoclonal antibodies. However, the impression is that we are quite adamant about asking for efficacy trials for monoclonal antibodies. The truth is that there is already an open door for not doing that. Next slide, please. So one of the exceptions that is unfortunately not too often applicable for monoclonal antibodies is the case where we have meaningful PD markers that can serve as a surrogate for efficacy. And if you wanted to discuss a specific PD markers, we strongly would recommend you do that in the European scientific advice procedure if they are not directly stated in the product-specific guideline already. And the second reason is you could come up with a comprehensive and meaningful quality comparability, and that could make you eligible for a tailored clinical approach. Next slide, please. How is this evaluated, whether the quality program is comprehensive enough to allow for that? We have a specific procedure for that. That's the tailored scientific advice. This actually is a special form of scientific advice that allows for pre-assessment. It has been out there for a few years, and to be frank, we've had mixed feedback about that, because only in very rare cases sufficient quality data is already available before the initiation of the clinical program. So we will review this and see whether we can tweak that process a little to make it potentially more useful for applicants and developers. Next slide, please. However, I can already name two examples of biosimilar monoclonal antibodies that have received a positive opinion in Europe and who did not have a full scope clinical efficacy trial. And I have to be fair, this was not because they discussed a specific this was rather caused by feasibility reasons, because this monoclonal antibody is only authorized for an orphan disease where it was not feasible to conduct a fully grown efficacy trial. So we went with rather small PD trials, PD and safety, and managed to assess biosimilarity based on that, together with quality and preclinical program. So one could argue that there are already two biosimilar monoclonal antibodies authorized in Europe based on a tailored approach, driven by feasibility. One has to be fair. Next slide, please. So what are our plans? In the Biosimilar Working Party, what we are doing right now is we are drafting a concept paper that should outline high-level principles regarding a potential tailored clinical approach. This is going to be for monoclonals. And our plan is to have a draft be presented this year to CHMP. So this is not the publication date. I'm sure there are going to be a lot of discussions centered around that. And another thing I have to be entirely frank about is this high-level concept paper saying this is in general possible. This is not the big change yet, because in certain circumstances it's possible already. And telling people that it is possible under specific circumstances, if your quality is especially comprehensive, that is just not the specific and concrete and solid kind of guidance that will change a whole lot. The second step is going to be that as soon that we have a version of that is published, we will start working on a so-called reflection paper, where we really want to go a bit more into detail and solidify principles how you could approach such a tailored clinical approach to waive efficacy trial based on a quality, on a comprehensive quality program. And it is not going to be easy, because in all the biosimilar conferences, I hear the same song over and over again. Let's just not do that anymore. But we have to lay down detailed principles that developers can rely on. And I'd like to put that actually out to the developers who are listening right now, because I have to say there are a few pioneers that are challenging us with novel approaches, driving the framework forward. But the large majority just claims clinical trials are useless and we shouldn't do them anymore. So I'd like to invite everyone to the discussion and come up not only with policy, but also with hard science. Thank you. That's everything from my side for now.
Thank you, Dr. Anwar. I would like to thank all the participants for their questions and answers. I just want to highlight that you submit a question in the Q&A. Can you please indicate who you are directing the question to, or let us know if it is for general response? Thank you. For our next speaker, Dr. Bradley Scott from Health Canada. Dr. Bradley is a senior clinical evaluator at the Biologics and Radiopharmaceutical Drugs Directorate within the Health Products and Foods branch at Health Canada. He received his Bachelor of Science degree in Biology from the University of Waterloo and his PhD in Cellular and Molecular Medicine from the University of Ottawa, where he conducted research for the identification of novel genetic factors involved in chemo and radiotherapy resistance. He joined Health Canada in 2009 and began working in the area of biologics regulation in 2011. He is actively involved in the review and authorization of biologics for use in hematology and oncology, as well as in the regulation of biosimilars. He has authored publications on the Canadian approach to the regulation of biosimilars and is an active member of the Biosimilar Working Group responsible for maintaining Health Canada's guidance relating to biosimilar drug submissions. Dr. Bradley, please take it away.
Good day. Thank you for the introduction, and just want to thank all the attendees for being here for the discussion today. Over the next 10 minutes, I'll speak about the Canadian experience in the review and authorization of biosimilars. I'll touch on the use of clinical efficacy studies in biosimilar submissions to support a demonstration of biosimilarity. And I'll also talk about some of the ongoing initiatives at Health Canada for biosimilars. So, sorry, do I advance the slides or? Okay. So I just wanted to start with a very brief history of biosimilar regulation in Canada. It was way back in 2005 when a sponsor first contacted us about products being developed with this concept. We formed our first working group in 2005, and that working group put out a fact sheet that sort of addressed the key points on how biosimilars were going to be regulated in the existing framework of the Food and Drugs Act and regulations. In 2008, the group issued a draft biosimilar guidance document, and very shortly after, Health Canada authorized their first biosimilar, which was a version of growth hormone. In 2010, we finalized that initial guidance document for what were then known as subsequent entry biologics. And fast forward to 2014, we received our first monoclonal antibody, which was a version of infliximab. The product did receive a notice of compliance. And experiences between 2010 and 2014 led to the reopening of the guidance document, which we did revise in 2016 to provide more information on what Health Canada expected within a biosimilar submission. At that time, we also adopted the name biosimilar biologic drugs, mainly to align with the nomenclature internationally. So fast forward to 2023, we have reviewed numerous biosimilars at this point, and we are again taking a look at our guidance document, and we'll be considering revisions. Next slide, please. So over this time period of approximately 15, 16 years, we have had approximately 68 new drug submissions for biosimilar products. We've issued 53 positive decisions. Most of those are unique products, but occasionally there are duplicates of a product that have different brand names. Over the period, we have had 10 submissions withdrawn from review, either because issues were identified during the review period with either the quality or the clinical side of review, or because they were part of a parallel filing where one product did receive a notice of compliance. We also have 5 products that are on patent hold, which means that the review recommendations have been rendered, but a notice of compliance cannot be issued until the patents are addressed. Next slide, please. So as I said, we have 53 total products authorized at this moment, or sorry, as of summer 2023 in Canada. 16, those products represent 16 unique biologics. 6 of those are insulin products that represent 4 types of insulin. Adalimumab is the most commonly produced biosimilar. We have 8 products representing adalimumab. Bevacizumab, trastuzumab also have a number of different brands that have been produced. And of course, these are important products that are coming onto the market in Canada. They span indications from oncology to autoimmune disease to diabetes. And we are starting to see common use in Canada and uptake by patients and prescribers. Next slide. So with respect to our current guidance and what it has to say about clinical efficacy studies, it currently states that in most cases, a comparative clinical trial is important to rule out clinically meaningful differences in efficacy and safety. Currently, there is an exception to that, and we've heard a little bit about that already from the other regulators, that a clinical trial may not always be necessary, in particular where we can rely on a clinically relevant pharmacodynamic endpoint. And one point I wanted to make is that Health Canada is not typically engaged by industry at the design phase of a biosimilar program. We do expect that manufacturers should be aware of our current guidance, and when filing a submission to Health Canada, should fulfill the requirements that are laid out in our guidance document. However, Health Canada will consider the data as it's submitted and will determine its adequacy to meet the regulations during our review. Next slide. So just in preparation for this forum, we looked back at our biosimilar submissions. As I mentioned, we've had 53 authorizations to date. And it's clear that the vast majority of submissions that we've received have included a comparative efficacy study. And you can see from the figure on the right that's about in the area of about 50 products where a clinical efficacy study was submitted. But I also want to point out that these efficacy studies are often shorter term studies looking at surrogate outcomes such as overall response rate for the oncology biosimilars, which may differ from the studies that originally supported authorization for the innovator products. We did have a few biosimilars, I think 9 of them that were authorized having no clinical efficacy study looking at what could be considered a clinical outcome. However, these submissions typically add characterizable PD endpoints with clinical relevance, which is in line with our current guidance document. And you can see that 5 of these were enoxaparins, where anti-factor 10A activity was looked at in combination with a number of other PD endpoints. We have 2 filgrastim products looking at absolute neutrophil counts, and also another pegfilgrastim product looking at the same, as well as an insulin product that relied solely on glucose infusion rates measured in a euglycemic clamp study. Next slide. So during the time reviewing the biosimilar submissions, we've often raised questions about the ability of clinical efficacy studies to detect differences. Some of our main concerns related to the sensitivity of clinical efficacies to observed differences versus what could be considered residual uncertainty related to potential undetected differences. And we've questioned whether it's reasonable that we should expect a clinical study to detect differences where no significant differences are seen in physical, chemical, or biological assays. We also have raised concerns about the setting of equivalence margins, which depend on prior study data. That data can often be limited and dated, and there can be difficulties with result interpretation when results differ from what might have been expected based on prior data. There's obvious concerns with reporting on multiple outcomes, including primary plus multiple secondary and exploratory endpoints. And this can lead to unwarranted concerns regarding biosimilarity, despite the known limitations with looking at multiple endpoints where multiplicity is not adequately addressed. And of course, we do expect that when we're looking at multiple endpoints, including safety and immunogenicity, that we will observe numerical differences. And these can be difficult to interpret, difficult to make decisions as to how these should affect an authorization. And they may also have effects on the perception of biosimilars in the healthcare professional world and for patients as well. Next slide. So I just want to leave you with a few updates from Health Canada on our currently ongoing biosimilar activities. We've recently updated our labeling expectations for biosimilar products, advising that comparative studies are no longer expected to appear in our product monographs. And I've linked to the policy statement on our website in the presentation. This will bring our product monograph into alignment with the European SMPC and the American USPI, where it is not common practice to include the results of comparative clinical efficacy studies. As I mentioned at the beginning of the talk, we have now reconvened our working group to open up our guidance document. And in the spirit of this meeting, we are going to be exploring whether there is a continued need for clinical efficacy studies. And we do expect to issue a draft of this revised guidance. Likely it will be early in 2024, after which we will solicit comments from stakeholders. And of course, we will consider those and address those before adopting a finalized revision. So next slide. Yeah. So thank you. Contact info is here, and look forward to hearing your questions later on.
Thank you, Dr. Bradley. Our next speaker is Dr. Ryusuke Kuribayashi from the PMDA Japan. Ryusuke is a Deputy Review Director at the Office of Cellular and Tissue-Based Products at the Pharmaceuticals and Medical Devices Agency since last year. Currently, he is responsible for the review of biosimilars and the quality of new biopharmaceuticals as well. Before that, he was in charge of the review of generics in the Office of Generic Drugs from 2013 till 2022. Before that, he served as a researcher at the Division of Biological Chemistry and Biologicals at National Institute of Health Sciences to engage in analytical research on biopharmaceuticals from 2010 through 2012. Before that, he served as a reviewer within the Office of New Drugs at the PMDA from 2005 through 2010. As other activities, he is a member of the IPRP Biosimilars Working Group and also a member of the ICH M30. Dr. Ryusuke, please go ahead.
Thank you for introduction. Can you hear me?
Yes, we can. Please go ahead.
Oh, thank you. Good morning, good afternoon, and good evening. My name is Ryosuke Kuribayashi. Next slide, please. Oh, thank you. My name is Ryosuke Kuribayashi from PMDA. Thank you for giving this opportunity to share our experience of biosimilar approval in Japan. Today, I would like to speak the regulatory experience and consideration to date from PMDA. Next slide, please. Here is my presentation outline. First, I will explain the history of biosimilars in Japan based on the notified guidelines and the number of approved products. Next, I will introduce the approved biosimilar programs, which includes a comparative efficacy study, a summary of learnings. Third, I will present approved biosimilar programs, which did not include a CES, and why one was not requested. Finally, I will talk about any post-marketing real-world learnings. Next slide, please. This slide shows the guideline and Q&As related to biosimilars in Japan and the year of approval of each biosimilar. The upper side shows the guidelines and Q&As, and the lower side shows the names of active ingredients and the year of approval of each product. After several revisions, the guidelines and Q&A for biosimilars in Japan were last updated in 2020. Somatropin biosimilar injection developed by Sanz is the first biosimilar approved in 2009. 32 biosimilars have been approved as the 2022. Antibodies and fusion proteins already account for more than half of biosimilars. Next slide, please. This slide shows a summary of clinical data package of biosimilars in Japan. As mentioned before that, 32 biosimilars have been approved as of 2022. 23 biosimilars were approved based on comparative analytical studies, a comparative PK study, and a CES. 7 biosimilars were approved based on comparative analytical studies and a comparative PK/PD study. In other words, 7 biosimilars were approved without CES. 1 biosimilar was approved based on comparative analytical studies, a comparative PK study, and a PD study. This is also approved without CES. Finally, 1 biosimilar was approved based on comparative analytical studies and CES. In other words, this is approved without a comparative PK study. Next slide. This slide shows a description of Japanese biosimilar guideline regarding the requirement of CES. As our guideline mentions that it can be challenging to demonstrate the comparability based on analytical studies only. On the other hand, our guideline mentioned where PK and or PD studies are sufficient to assure comparability in the clinical endpoint of interest. Additional clinical studies to evaluate efficacy might be omitted. As described, that our guideline does not describe that a CES is mandatory for biosimilar development. We have a regulatory flexibility of not requesting a CES. Next slide, please. Next, I would like to share the reason which did not include the CES. First example are filgrastim and insulin BS versions. These 7 biosimilars were approved without a CES because PD marker, that is a validated surrogate marker for clinical efficacy, are available for these products. This case is in line with Japanese biosimilar guideline. Next slide, please. Next example is agarcitas beta BS version. This biosimilar is approved without a CES because a biosimilar of agarcitas beta is used against the patients of Fabry disease, which was a rare disease. A sponsor did not conduct a CES and also a 2-armed comparative parallel designed PD study due to feasibility, disease property, and the mechanism of action. That is, this product is used as the enzyme replacement therapy. Therefore, the sponsor conducted a PD study of single-arm switch therapy from originator to biosimilar. The detailed study design is available in the public article. Next slide, please. Finally, I would like to share our experience of post-marketing surveillance for biosimilar. First of all, Japanese regulations require the implementation of risk management plan as the approval condition based on the domestic notification. Teriparatide BS and infliximab BS, 2 products were evaluated the safety and efficacy in post-marketing surveillance. Then, in this year, approval condition of 2 products was removed because the approval condition was satisfied. And also, no safety concern more than originators is found through PMS of the biosimilars so far. Next slide. Thank you for your attention. That's all.
Thank you, Dr. Ryusoke. Our next speaker is from the MFTS Republic of Korea, Dr. Woo Young-oh. Mr. Oh is Acting Director of Recombinant Protein Products Division at the Ministry of Food and Drug Safety in Korea. He has been responsible for reviewing non-clinical, clinical, and quality documents of recombinant protein products since 2022. Before that, he had worked as a senior scientific reviewer in drug evaluation department, followed by his 5-year stint as a researcher in division of clinical research. In 2001, he joined former MFTS KFTA and started his career as a scientific officer. He has worked on the development of many guidelines on the evaluation of drug efficacy and safety. Dr. Oh, please go ahead.
Yeah, good day to you all. Yeah. Next slide, please. Next slide. Yes, so I'm going to present Korea MFDS regulation on the biosimilar approval, confirmatory clinical study, and the post-marketing surveillance in Korea. As we are well aware, a biosimilar is a biological product whose quality, non-clinical, and clinical comparability has been demonstrated against its reference product, and all the clinical indications that approved for the reference product can be extrapolated to the biosimilar based on its equivalence to the reference product. Next slide, please. Let me explain Korea's laws and regulation applied to the regulation of biosimilars. They are the Pharmaceutical Affairs Act, enforcement regulation on the safety of drugs, and regulation on review and authorization of biological products. These regulations state that for biosimilars, clinical study data should be provided, which can demonstrate the compatibility between the biosimilar and its reference product. And in principle, these clinical data should be those generated from comparative studies. Next, please. The requirement to be submit for marketing approval of the biosimilar and other relevant information can be found in regulation on review and authorization of biological products. MFD's biosimilar guidance document has been revised since their publication starting from 2009 to be aligned with the revised WHO biosimilar guideline. You also have 6 product-specific guidelines, include the guideline on somatropin biosimilar. Next, please. Now, let me share with you the gist of the clinical data consideration in our guideline for biosimilars. The guideline describes that PK and PD compatibility clinical studies are requirements for biosimilars. However, if sufficient evidence demonstrating equivalency is obtained from other compatibility exercises, such as qualitative and clinical comparative PD studies, then comparative efficacy studies may not be necessary. Case in point are biosimilar, GCSF, insulin, and teriparatide, for which comparable clinical studies were waived. Next slide, please. These slides show the following 3 slides. Let's look at the current landscape of the approval biosimilar in Korea. Biosimilar developer in Korea was Celltron, Samsung Bioepis, LG Chem Limited, Jongundang, and Panjen. Celltrion Remsima is Korea's first biosimilar ever approved in 2012. Since then, as of June 2023, a total of 15 biosimilar products has been approved in Korea. You can see the table, the active ingredient of each product and their respective approved dates by MFDs, FDA, and EMA. Next slide, please. Next slide, please. Biosimilar imported from global companies, there are 8 products, including Saizen Korea Cytropin A that contain somatropin as inactive ingredient. Next slide, please. This slide shows comparative efficacy study of biosimilar in Korea. A total of 23 biosimilar has been approved in Korea. Taking a look at the phase 3 clinical efficacy studies conducted for this biosimilar, one comparative efficacy study was conducted for each product, and the sample size was around 100 to 900 subjects, where equivalence over non-inferiority of the biosimilar to its reference product was confirmed. Next slide, please. Out of the 23 approved biosimilar, there is 1 product approved without phase 3 clinical trial data, and also 1 product that was approved even though it has failed its phase 3 compatibility efficacy study. For the rest of the 21 biosimilar, phase 3 clinical data was submitted, including insulin glycine that could have been proceeded without phase 3 clinical study as described in MFD's guideline, and its equivalence or non-inferiority to the reference product was demonstrated. First, let me talk about the biosimilar that was approved without demonstration of equivalence in the compatibility clinical study. similar difference in complete response rate between the biosimilar and the active control was not within the pre-specified equivalence margin. However, in terms of quality aspect, there was the ADCC variation in the reference product that was used in the phase 3 clinical trial, which could likely impact the efficacy outcome. So, analysis was conducted excluding patients given the ADCC variable reference product, and the results showed that the complete response rate difference fell within the equivalence margin. In addition, the comparison of EFS or OS conducted in the following year confirmed the similarity of the biosimilar to the reference product, which finally led to the approval of the product. Let's see the case there. Comparative efficacy study were waived. For biosimilar teriparatide, randomized double-blind single-dose crossover PK comparative clinical studies were conducted, and the pre-specified equivalence margin was fulfilled, based on which this product was approved as biosimilar. Considering the characteristics of the teriparatide, such as its simple structure, low molecular weight, absence of the post-translational glycosylation, comparability efficacy study was not absolutely necessary. Next slide, please. Let's move on to post-marketing safety management of biosimilars. For biosimilar, the examination should be conducted for a 4 years period following approval as prescribed by enforcement regulation on the safety of drug. The purpose of the examination is to investigate the occurrence of new adverse events and the factors that affect the safety and the effectiveness of the marketed biosimilars, which have not been identified yet during the pre-marketing assessment process. Next slide, please. All of the 23 approved biosimilar was subject to the requirements of the examination, and 9 out of 23 has completed the examination. After the completion of the examination, incidence rate of the adverse event, serious AE, and unexpected AEs that have been identified incorporated into the precaution in use. The sample size lead for the examination was around 180 to 1,400, with the means of 553. No emergent safety concern has been identified for the biosimilar that have completed the examination. Next slide, please. Here are conclusion and the key takeaway from our experience. Biosimilar in which comparative efficacy study was conducted accepted teriparatide, considering its structure and characteristics properties. For 23 product approved as a biosimilar, post-marketing the examination has been completed or is underway, but there have been no new safety concerns. This is all I have prepared for today's presentation. Thank you for your attention.
Thank you, Dr. O. Our next speaker is Dr. Stacy Ritchie of the FDA, United States of America. For the past 18 years, Dr. Ritchie's work at the FDA has focused on the scientific and regulatory review of therapeutic protein products. In her current role as Director of the Scientific Review Staff in the Office of Therapeutic Biologics and Biosimilars, Dr. Ritchie leads a multidisciplinary team of scientists, clinicians, pharmacists, and project managers who oversee the review of biosimilar and interchangeable products at all stages of development, and to advance biosimilar policy and scientific standards by conducting regulatory science research, facilitating scientific dialogue within FDA and through stakeholder engagement, and developing and contributing to guidance and rulemaking, and providing educational and training opportunities. Prior to joining the FDA in 2005, Dr. Ritchie completed postdoctoral research at the University of Pennsylvania, received her Doctor of Science degree from Tulane University, and Master of Engineering and Bachelor of Science degrees from Cornell University. Dr. Stacy, please go ahead.
Thank you so much. Good morning. Next, can you bring up my slides? Thank you. Good morning, good afternoon, and good evening. It's an honor to be part of this workshop with so many of our international colleagues and stakeholders. Thank you for joining us today, and to everyone who helped to organize the workshop. I thought it would be helpful to set the stage for our discussions of the utility of comparative clinical efficacy studies for biosimilars over the next couple of days if we consider these questions. Where are we now and how did we get here? Where do we want to go and how to get there? And for my brief presentation, I'd like to walk through some of the factors I think can help answer these questions. But let's first briefly touch on where we are today. Next slide, please. So in the US, FDA has approved 42 biosimilars to 12 different reference products, 4 of which are interchangeable. These 42 products are approved to treat a wide range of diseases and conditions across many different therapeutic areas, including the side effects of chemotherapy, hematologic malignancies and other cancers, autoimmune diseases such as rheumatoid arthritis, psoriasis, Crohn's disease, diabetes, multiple sclerosis, and many others, and the list continues to grow. A brief aside about interchangeability in the US, this is a legal designation that a manufacturer can request for their biosimilar, and it means that the biosimilar can be substituted for the reference product at the pharmacy without consulting the healthcare prescriber, and a process which is subject to state law. A healthcare provider can prescribe a biosimilar to their patients at any time, whether they previously received the reference product or another biosimilar to the reference product, or if they have never been treated for their disease before. Next slide. To support approval of a biosimilar, a manufacturer must provide data sufficient to demonstrate the biosimilar is highly similar to the reference product and that there are no clinically meaningful differences between the two. So this graphic sort of represents where we are today. The data supporting the approval of the 42 biosimilars all included detailed analytical characterizations from a comprehensive battery of physicochemical and in vitro biological tests, confirming the biosimilars are highly similar to the reference products in all important and relevant structural and functional attributes. The comparative data used to address that there are no clinically meaningful differences between the products has varied, but can be divided into 3 general categories. And starting on the left, a clinical study comparing pharmacokinetic and pharmacodynamic endpoints using statistical criteria would be sufficient in the case of insulin products. So no efficacy data. For biosimilars to the 4 reference products in the middle, studies evaluating PK and PD in combination with a descriptive comparison of immunogenicity and safety was sufficient. And for these 7 reference products where there weren't suitable PD biomarkers available, a comparative efficacy study in patients was conducted to establish statistical evidence that the biosimilar is neither inferior nor superior to the reference product. Next slide, please. I think it's helpful to understand where we are today in terms of the clinical efficacy data expectations when we consider the timeline of key milestones in FDA's regulation of biosimilars. It's been almost 14 years since Congress granted FDA the authority to approve biosimilars as part of the Biosimilar Price Competition and Innovation Act, which described that an application for a proposed biosimilar must include the types of data illustrated in the pyramid here. So analytical studies, animal studies, and a clinical study or studies, including and those studies should include an assessment of PK or PD and immunogenicity. So while the statutory language of the Act was very prescriptive about the types of data to include in an application, the Act also provides FDA with flexibility to determine that any of these specified data elements, analytical, animal, or clinical, were necessary. And this essentially means that FDA could determine which data are needed to support a demonstration of biosimilarity. It was only a couple of years later when FDA published the seminal guidance with recommendations about the approach sponsors should use when collecting data necessary to demonstrate biosimilarity. The approach recommended was that sponsors should generate comparative data between the biosimilar and the reference product in a stepwise fashion that begins with a foundation of detailed analytical characterization data, comparing structural and functional attributes of the two products, moving on to animal studies if necessary, and then conducting a comparative clinical pharmacology study and any additional clinical studies necessary, where at each step, the type and amount of data needed beyond comparative analytical data would focus on addressing any remaining uncertainty about biosimilarities that might exist. In these guidance documents, FDA recommended that sponsors should conduct comparative human PK and PD study if there is a relevant PD biomarker available, and compare the clinical immunogenicity of their product to the reference product in an appropriately sensitive population. FDA acknowledged early on that PK and PD parameters are generally more sensitive than clinical efficacy endpoints in assessing the similarity of two products. FDA also recommended, when there weren't relevant or suitable PD biomarkers available, additional comparative clinical studies conducted to address any residual uncertainty about biosimilarity should be designed to establish statistical evidence that the biosimilar is neither inferior nor superior to the reference product. It's notable that this recommended approach was developed before FDA had formally reviewed any biosimilar applications under its new authority, as the first biosimilar was approved a few years after the recommendations were published. Next slide. So while on a conceptual level, the stepwise approach was quite reasonable, the practical aspects of designing and implementing a biosimilar development program has a timeline that doesn't match up very well with the recommended stepwise approach, which on a fundamental level is based on the expectation that the comparative analytical assessment is sufficiently complete before a developer would design and initiate a clinical study or studies to address the residual uncertainty that might remain based on differences observed in the analytical comparisons. So for example, considering the product development timeline is a process that involves characterizing multiple lots of the reference product that have to be collected over a sufficient duration to capture the expected variation in post-translational modifications and other microheterogeneity inherent in therapeutic proteins. And that's an ongoing process that's done in tandem with piloting and scaling up manufacturing and control processes to generate clinical and commercial lots of the proposed biosimilar, all while accruing data needed for the comparative analytical assessment. During these activities, developers meet with the various international regulators to gain agreement on the analytical and clinical data needed to support filing applications for their products, and specific details of the clinical study protocols and associated statistical analysis plans should be clearly understood by both developers and regulators before the initiation of any clinical study. So from a logistical perspective, that likely have major financial considerations. The need to have agreement on the clinical studies early in the development process is important to the success of the biosimilar program. And it's after discussions with regulators that the comparative clinical studies are initiated and completed all along in tandem with completing the comparative analytical assessment and collecting any other data necessary to support the application. Next slide. So returning to the question of where we are now in terms of clinical data expectations with statistical evaluations of clinical efficacy, the practical aspects of biosimilar development don't really lend themselves to a stepwise approach because it assumes the comparative analytical data package can be sufficiently complete before making decisions about the clinical data needed. I believe other contributing factors to where we are now in terms of clinical data efficacy expectations. Can you go back? Can you go back one slide, please? Thank you. So both regulators and developers, had to make decisions about what clinical data would be needed to assure themselves and prescribers that biosimilars are just as safe and effective as their reference product for any of the reference products' approved indications. And this was a pretty tall order given the general lack of experience using biosimilars across clinical specialties, as well as the newness of the biosimilar regulatory pathway, which has its own unique terminology and scientific considerations that are different from the more familiar abbreviated pathway for generic drugs. I also think that a collective unfamiliarity with the comparative analytical data serving as the foundation of a biosimilar application and theoretical concerns about their safety and effectiveness were also major contributing factors to expectations about clinical efficacy studies. Having studies designed based on hypothesis testing using statistical equivalence criteria would be familiar and help to increase confidence in biosimilars for both the regulators reviewing and approving biosimilars and to healthcare providers and their patients. So by default, in situations where reliable pharmacodynamic biomarkers weren't available, expectations from both sponsors and regulators was to conduct comparative efficacy studies, as these are the gold standard for demonstrating safety and effectiveness in the context of new drug development for products that are seeking to be superior than, for a comparative clinical study in the context of a new drug development program. Okay, so given where we are now with over 40 approved biosimilars and more on the way, where do we want to go and how can we get there? Next slide. Here I've begun a list of some ways that we, meaning both regulators and manufacturers, can collectively help to streamline clinical data expectations to support a demonstration of biosimilarity. First, I think it's important that we work towards increasing confidence in the comparative analytical assessment by focusing efforts to explain why robust structural and functional analytical comparisons that demonstrate products are highly similar can provide assurance, can you go back please, that a biosimilar will have the same clinical performance as its reference product. As part of this educational effort, we also need to highlight the evidence which shows that clinical endpoints simply are not as sensitive as analytical data to detect differences between products. Another important effort is to develop risk-based criteria to justify when a more limited clinical assessment, for example, a single-dose PK study in healthy subjects, is sufficient to complement the comparative analytical data needed to demonstrate biosimilarity. And then once there's an agreed upon approach for how to streamline clinical data expectations, and then we as regulators can provide updates to our guidances or guidelines as appropriate. And that's the end of my talk. Thank you.
Thank you, Dr. Stacy. Okay, now it's our time for the panel discussion. I want to thank the panelists as well as the participants for their contribution to the questions and answers. MHRA, Marie-Christine, WHO Dr. Ema Kang, Ema, Dr. Reed Anwar, From Health Canada, Dr. Bradley Scott. From PMDA, Dr. Yusoki. From FDS, Dr. Roh. And from USFDA, Dr. Stacy Ritchie. We have plenty of questions, so I'll try to be fast. First question to the panelists. What do you think are the common reasons clinical efficacy studies have been utilized widely in biosimilar development programs so far? Why do you think that is?
Well, I tried to walk through the reasons I think that they are utilized, and really I think it's a historical, based in historical reasons for the unfamiliarity with biosimilars. It's the unique considerations that exist for therapeutic proteins, which are, there's no expectation that the reference product will be a homogeneous mixture, right? It has microheterogeneity, and so there for that basis, the scientific data that are needed to demonstrate the products are similar is different, and it has taken time for people to gain experience with collecting those data, understanding those data, and having confidence in those data, because the default generally is that the clinical data are the gold standard for demonstrating a product is safe and effective, when in fact, if you have a copy of a product, right? I mean, biosimilars are essentially copies of old drugs. If the copy is something that is highly similar, then where do the questions remain about, how could it have a different clinical performance?
Thank you, Dr. Stacy. We have a question for Dr. Marie-Christine, MHRA. Dr. Marie, can you hear me?
Yes.
A question says that in your experience, are clinical efficacy studies results being used to resolve residual uncertainty arising from comparative analytical assessment, or are clinical efficacy studies typically considered on their own? What do you think?
This should be addressed to my EMEA colleague, because I don't think that any residual uncertainty on the quality from the quality assessment can be resolved by a clinical trial, which is completely insensitive. So if there are uncertainty, if there are differences, they have to be resolved either by reference to the literature, whether they may have a clinical impact, or additional biological tests. And finally, if it's not possible, then the manufacturer should work better to make a more similar molecule, rather than to try to test it in 400 oncology patients to show that it doesn't seem to make any difference. So for me, it is clear that it can't resolve uncertainty. And typically, I think in the past, and this is for historical reason, because we didn't know in the beginning, they were just made as the last step of the comparability exercise to confirm the biosimilarity. And I'm stopping there.
Thank you, Dr. Mari. Dr. Rene Anwar, do you want to comment on that, please?
So, first of all, I believe that is exactly how the clinical trials are being used now. And I believe that if we would have stopped earlier in doing them, many of those uncertainties that wouldn't have mattered at all in the clinical performance would have been showstoppers, because there's just no information out there how they would translate into the clinical profile or anything. And then we would have had, I mean, it's easy to say the applicant should work better, that would be a failed marketing authorization. I believe we should go that route in omitting the trials, but I think the future is that the applicants must prospectively define what quality attributes account for what on the clinical profile, in order that we don't even have uncertainty when it comes to the assessment. For now, I believe, yes, this is how we use it. We encounter some kind of uncertainty that cannot be too grave, because the clinical trial cannot rescue that. But if we have something that might or might not be a problem, usually the outcome is that it doesn't matter. It is not clinically relevant, and this is how we use it right now.
Thank you, René. We have a question to Dr. Ryusoki. It says that since comparative studies, PK or efficacy studies, are focused on showing similarity between products, can you comment please on the requirements to conduct them in Japanese subjects, or if there is flexibility to accept global or foreign patient data? In other words, no need to do specific clinical study in Japanese population. Dr. Ryusoki, please, what do you think?
This is Yosuke. Can you hear me?
Yes, we can. Please go ahead.
Yeah, thank you for question, but this question is out of scope today's meeting. So first, I would like to ask you to check the Japanese biosimilar guideline.
Okay, we'll receive. Yeah, we have a question to the panel. It says, what information can sponsors provide that would be most helpful in supporting a determination that a clinical efficacy study is not needed for a given development program? Please, anyone from the panel, feel free to answer. Go ahead.
Who wants to go first?
I guess I'll take a shot at that.
Go ahead, please.
I think this is more of a paradigm shift than a specific biosimilar program where a particular manufacturer should address this. I think what regulators need from industry is solid data on the predictive capabilities of analytical data to predict clinical safety and efficacy outcomes, so that we know when we have a program showing the two products are biosimilar, that we don't expect any differences in clinical performance. One of the key areas that is always cited is immunogenicity. And I think right now, to the best of my knowledge, is that we don't have great information on how we can predict immunogenicity based on the bioanalytical and how that reflects in the clinic. So if that data is generated and can be provided to us to support our decision making, that will go a long way.
And to add on to what Bradley just said, if a lot is known about the immunogenicity of the reference products. So if there are analytical differences that are not expected to impact immunogenicity, then what would the basis of that be? Like, how do we provide assurance that the minor analytical differences, which exist, as we know, in the reference product as well, will not result in any differences in endpoints such as immunogenicity? Like, where are the clinical data? And if we do collect clinical data, what's the expectation that those data will address the concern that we have about an analytical difference that we otherwise can't interpret? So there's certainly some analytical differences that are observed and really are going to be too great for to meet the standard of highly similar. And in that case, as Marie-Christine mentioned, then then a change in the manufacturing process would be the next step, right? To align more closely to the reference product. Clinical data aren't going to rescue that difference, even if there's no, they show equivalence in all relevant endpoints.
Thank you, Stacey. So are there any efforts made to come to a harmonized understanding of what is residual uncertainty is meant and how it can be quantified?
Does your question rather refer to harmonization of our approaches, or how we can address that problem in principle?
How we can address it in principle? Yeah.
I think it's very difficult to quantify residual uncertainty. I think that it would be dependent on a program, on the molecule, on the data. There isn't a cookie cutter way to assess residual uncertainty. And I cannot stress enough what is being already talked about by the other panelists, that the analytical data are really the basis. And the most important thing is to demonstrate that the products are highly similar. It's so far what we've seen are very little differences, and and those very little differences cannot really be addressed by clinical studies. But once again, residual uncertainty is not a mathematical equation. It's determined case by case.
Thank you, Manuela.
To add on that, I don't think the question is too much residual uncertainty, but it's rather how is a comprehensive quality comparability program set up? What does that even mean? And what is saying prospectively, what is success and what isn't? And I believe that is where a lot of work has to go into from both sides, actually. We need to consider for different molecules and different attributes what the impact of a difference there is, how big that difference can be, how often that occurs, what's the best way to compare. Is it an attribute where we should refer to inferential statistic, or is it an attribute where that doesn't make sense at all? And this is the work that future biosimilar developers will have to do, and they will have to come up with a very sound transdisciplinary quality dossier that allows to make an assessment of clinical comparability without having a clinical trial. And this is, in my opinion, where the development must happen. The regulator's duty here is to lay out principles, and the developer's duty is to follow those and come up with a very sound quality comparability that is grouped around clinical attributes such as efficacy, safety, immunogenicity, and that allows for translating those results into the clinic.
Thank you, Renee. We have a question to Dr. Bielski, Scott, and Dr. Anwar. He's asking about whether anyone would consider waiving bioequivalence studies, or whether the focus is on comparative efficacy studies. What do you think?
What's the question? Whether...
Yes, sorry, can you repeat it? Because I'm not sure I understood.
It says, can we ask whether anyone would consider waiving bioequivalence studies, or whether the focus is on comparative efficacy studies?
I am surprised by this question because I thought there was one question which was, can you go even further and waive the PK study, but obviously not replace it with an efficacy study, because as our position is that in most cases, not all cases, there may be cases where an efficacy study is required, but in most cases, if you can derive the efficacy and safety and immunogenicity from the analytical exercise, and very importantly, the mechanism of action. Obviously, if you don't know the mechanism of action of the reference product, then we don't think that this is possible. So, but it's up to now, we've not seen that there is a reference product where there is no idea about its mechanism of action. But if that's the case, that's the approach. And but it's true that you can wonder whether, for example, for intravitreal products, what should we do if a parenteral, an IV or subcut product doesn't exist and is it's only an intravitreal product? What do we do? Would the analytical exercise only be sufficient? And if it's a really wonderful comparability exercise, maybe we would think about waiving an intravitreal study, because that would be in the end an efficacy study, or whether we would only look at some safety data. So in order not to go directly from nothing at all, no clinical data. This may be a jump which is a bit, which may be a bit too big, but it may be then in that case we would agree that safety data on the biosimilar only would be appropriate, just to check whether the product is reasonably safe. So that was the question, whether we could even go further than where we are now, saying that in most cases we don't think that an efficacy trial is necessary, is to go even further and say we don't need a PK trial. But this will really, at the moment, it's not on the agenda, and may happen for very specific cases.
So I agree with everything Dr. Bielski just said. I think the only case where we have considered not having a PK study is also for a ranibizumab ophthalmologic ocular products, where they're injected into the eye. And I don't think, I did mention that we are revising our guidance document in the process of that, and waiving PK studies outright is not one of our considerations currently.
I mean, this would take biosimilars, the requirement for biosimilar developments, below that of most generics, and I don't think that is realistically, with possibly a few future exceptions.
If they are IV, it's the generic don't have bioequivalence.
Yeah.
So for IV product, that might be really the question. But at the moment, we still think that we want immunogenicity from the PK study, the safety, local safety, infusion reactions. So we still have some data in human from this comparative PK study.
Dr. Hina Tang, would you like to comment on that, please? Dr. Hina?
No, I don't have a comment on the question, but I just answered to the question addressed in the Q&A chat box regarding the third question, the third bullet point question. Yeah, I provided some list of factors could influence to the decision on the need of efficacy data requirement. Yeah, I'm not sure whether all audience has the interest in the factors, because in the revised WHO guideline, we provide a list of factors could influence to the decision of the need of clinical efficacy trial. For example, how well the biosimilar can be characterized, and the second one is the availability of like a sensitive analytical method, for not only for the analytical, also the functional characterization, and the degree of analytical and functional similarity between biosimilar and reference product, and whether any relevant PD parameter is existed, and the degree of understanding of mechanism of actions and knowledge of the potential unwanted immunogenicity, yeah, and the information from the impurity profile. Yeah, that's the answer I provided.
Thank you, Dr. Hina. So, would any of the panelists answer bullet point number 3 on screen? What information can sponsors provide that would be most helpful in supporting a determination that a CSS is not intended for a given development program? Please go ahead. Anyone would like to volunteer to go ahead?
I've commented on that, so I will.
I think we've commented on that, but you know, under Canada's current guidance, of course, the exception to a clinical efficacy study right now is to have a pharmacodynamic endpoint that's clinically relevant that can be compared in a PK/PD study. Going forward, those requirements could change, but that is the exception right now.
I think the other panelists have also alluded to this, but if their analytical program really provides the assurance that their product will not have any structural differences, you know, in post-translational modifications. I believe one of the questions in the chat we're asking, like, what type of differences can be observed? These are, you know, the protein variants are things that can include different glycoforms that are attached to a protein that are generated during the, you know, the processing of the protein within the mammalian cells. Other types of chemical modifications, and so these variants may exist. There may be, you know, minor differences between the biosimilar and the reference product in that respect. So what is the justification from the manufacturer's perspective that those very minor differences won't have a clinical impact? If, you know, if a robust scientific justification is brought forward, you know, that's certainly going to be an important consideration about the question of, okay, if you want to propose not collecting comparative efficacy data or pharmacodynamic data, right? Because it's at this point within the FDA, it's an either or situation, then that can be certainly considered. But it's at this point, it's really incumbent upon the biosimilar developer to make that justification.
Thank you, Dr. Stacy. We have a question to AMA. It says that my question is for EMA that still states that the default is clinical trial and the FDA that maintains similar status. Can both agencies agree with MHRA that the developer should submit reasons for not conducting efficacy testing? Dr. Rene, please go ahead. What do you think?
So that possibility is already open. We do have the tailored scientific advice. In that scope, such a proposal can be discussed. We will also try and revise that to make it a bit more adjusted to the reality of the developers, but that's already possible. Yep.
I have a question to the panel. It says that given that most biosimilar development programs are global, how can sponsors and regulators best seek alignment across regulatory agencies?
I think the IPRP Biosimilars Working Group is a really good forum for us to continue these discussions and seek alignment on approaches that we can agree on for streamlining the clinical data expectations and helping to really be deliberative about the types of clinical data that would be needed in given situations as far as we understand now. I mean, the types of biological products for which biosimilars can be manufactured is growing, and there are certainly types that we haven't considered at least at the FDA. So that those may evolve over time as well. But I think given the current landscape of the type of recombinant proteins generated from DNA technology that can be highly purified, I think there's a lot that we do understand, and so maybe we can make progress on that front.
Thank you,
Can I just make a comment? I noticed a comment in the chat that based on today's session, clinical efficacy study is unnecessary if there's a surrogate PD and a PK study can be performed, but otherwise a CES is always necessary. And I just wanted to clarify from the Canadian perspective that, yes, it's true that this is what our guidance says in terms of the clinical requirements, that a CES study could be waived in the case that there's a PD surrogate. But just want to clarify that, it is guidance, and guidance is not binding. Of course, Health Canada would be open to discussing with sponsors other ways to demonstrate biosimilarity. So I didn't want to imply earlier that we are completely closed off to the idea of other options.
Thank you, Dr. Bradley. We have a question to Dr. Ritchie. It says, what are the possible considerations for waiving of switching clinical study data requirements or interchangeability designation? Dr. Ritchie, please.
So this is a topic that's really outside of the discussion for today, but I mentioned there are 4 products that are currently approved as interchangeable. Only one of those had additional data beyond what was expected for the biosimilar approval. So the concerns around immunogenicity that may result from switching between the biosimilar, or the reference product and biosimilar, are what kind of drove the recommendation that's in our interchangeability guidance about conducting a switching study. So when the concerns don't really apply to a given reference product class, then the switching study expectation is not important.
Okay. Well, question to the panel saying that following a stepwise approach, we demonstrate preliminary analytical similarity on the basis of pilot scale data of the proposed biosimilar. Can this preliminary data, if deemed highly similar, be considered sufficient to get endorsement for waiving phase 3 study?
I will start in general. Yes, but we'd have to look at the specific program and data. It's not the data alone, but also how it has been evaluated and whether the understanding of highly similar between the agency and the developers are aligned.
So what would it take, Dr. Rudin? Can you give more information, please, or more details? So if there is a manufacturer who would propose this, what would you advise to him or her?
Oh, to whom? Oh, okay, so again, sorry for repeating myself, but we have a process for that, which is the tailored scientific advice, where developers can submit the whole program as well as any quality data that they have at that time point, and then discuss whether that is sufficient to allow for a tailored clinical approach, which can, in theory, go as far as waiving a phase 3, an efficacy trial.
Thank you, Dr. Rene.
Sorry, this is Sarah. I just wanted to pull the standard questions off because I think people are getting distracted and there are a ton of questions in the Q&A. But I did want maybe Dr. Lacana, could you comment a little bit on sort of pilot scale versus later manufacturing scale up and what the challenges are in determining comparative analytics at that stage?
Thank you. You just read my mind. Yeah, there are issues when we're talking about preliminary analytical data. How preliminary are they? And as Dr. Yin mentioned, the scale is really important because we've seen not only in biosimilars, but also in standard program that scale up may really have an impact on the comparability of the product. So it is really important that the analytical comparability assessment, a decision is made in a product that is close to its final proposed marketing scale, commercial, because the scale up can really affect the comparability of the product, pre and post change product, and the similarity to the reference product. So again, preliminary is complicated. We looking at a product that is made at the 10-liter scale or 100-liter scale, and the proposed commercial scale is 10,000 liter, can be really complicated. So there are all these things that need to be taken into consideration when, approaching the agency. And we also have the ability to discuss with sponsor their development program. We have various type of meetings where a sponsor can submit different type of data, even very extensive data that we can provide advice to. So sponsor really should take advantage of their ability to communicate with FDA for their development program.
So just as a follow-up, and I apologize, Ali, for jumping in here, but I realize how many questions are coming in and how difficult it is to sort of keep track of what's coming in. Just to follow up on that, is there a way that regulators can think about if a product can achieve analytically highly similar, like what with the BLA submission? How can we think about our advice about comparative efficacy studies and whether or not they might be needed at an earlier stage? Can we sort of do more of a dichotomous kind of assessment where we think, okay, if you can reach highly similar with your BLA, then the clinical study might not be needed? Or do we even have enough information to make that kind of assessment early in development when folks are asking these clinical questions?
I think it will depend on the data that are provided early in development. You know, it's not that simple to make a prediction based on a limited set of data, what the product ultimately would look like. And as I mentioned before, especially if there is going to be a scale up or a large scale up from the data that we see early in development versus what we see late in development. It would be a risk for an applicant, but they would be welcome to say, okay, from my perspective, I have a very highly similar product and I will submit it to the agency and see what they say. It would be a risk, obviously, but if somebody is interested in taking it.
Does anybody else have any on the panel have any thoughts on that? It sort of goes to the stepwise approach that we've sort of historically taken, and how that timing doesn't really line up with development realities. Any thoughts on how we might be able to find an alternate kind of approach rather than make it stepwise?
The only way I see to prospectively define whether this is acceptable is to do that based on concepts, not on quality data, but how the quality program is being set up, what means success and what doesn't. And I believe developers need to focus on that to make a really strong case in what's a relevant difference there, what are the critical quality attributes. I know I keep repeating myself, but it's so important. And it's for many molecules, we are not there yet that everyone knows what those are. And yeah, the impact they might have, how realistic it is that that might cause a problem. All of those questions need to be answered. And again, in the end, I believe the result of this needs to be a clinical comparability without a clinical study. Just speaking from the European perspective, the people who decide on the marketing authorization, it's a clinical committee. So they need to thoroughly understand how that program, that submitted program, can account for a clinical trial. And I believe more than ever before, the challenge that needs to be addressed is to come up with really a transdisciplinary approach on quality.
By transdisciplinary approach, do you mean people of clinical disciplines need to get more comfortable with quality, or what exactly do you mean?
Actually, I mean, I think quality people on the developer side and on the regulator side as well, needs to get used to the novel larger weight that is put on that kind of data. And there needs to be, and I believe on both sides, closer collaboration between the disciplines in order to make that understandable, what is being planned on the quality level and what it finally means. And also, I think from the clinical or methodological side, let's put it like that, the information will need to flow to the quality in order to state for different attributes. Do we need inferential statistics there? It might be completely irrelevant for certain attributes. For others, it might make totally sense. And then there needs to be a prospectively oriented program that tells you, no matter what, that result means similar and that result means it's not similar. And that also, because we had the topic today, that strongly cuts down on residual uncertainty. And it sounds as this might be easy. I believe all of us have a lot of work before us to make anything close to that happen. But I believe this is the only way I see forward if finally those studies can be waived more routinely.
Anybody else, any other thoughts on how to sort of address kind of I mean, should it be that we're thinking about products that are high risk versus products that are low risk, and maybe limiting clinical studies to higher risk products, since it won't be that we have at the very beginning when we're being asked about clinical studies, we won't have a ton of analytical data, especially from higher manufacturing scales.
I guess, Sarah, my thoughts on that are that if manufacturers want to start seeing clinical efficacy studies being waived, then really they should think about following the stepwise approach that we've always advocated for. And doing so, they're going to have to provide quite a robust analytical package right up front so that these decisions can be made. Otherwise, it just defaults to the concurrent program where maybe the sponsor sees too much risk in only doing an analytical program on its own because it will cause delays. But I think that's what it takes. We need to see the information up front to make that decision, especially in this potential transitional period. At the end of the day, the onus is entirely on the sponsor to prove that their product is biosimilar to the reference product.
Thank you, Bradley. And we have a question for you, Sarah. It says it's about where do you see the harmonization and regulation for biosimilars going 5 or 10 years from now? What do you think? What are our thoughts on that?
You know, my hope is that we all want to make these programs as efficient as possible. The real question is going to be the details, right? And I think it's also going to be a question of there are so many folks who are involved in reviewing these programs that we have to get alignment within our agencies as well for all the folks who are you know, making these determinations and providing these recommendations, that they feel comfortable with what they're seeing that's not clinical data, right? I think clinical data makes the most sense in terms of the efficacy data is it's not sensitive, it's expensive. Companies have to start off very early. So it's a high cost in terms of time of development and cost of development. So obviously this is a big sort of goal. And my hope is that we get to a state where we're more comfortable going without comparative efficacy studies. But I do think there has to be some sort of framework for it, right? I think it's difficult to look back and just say, well, you know, on all the ones that we approved, the clinical studies weren't that helpful. To me, it's a little bit hard to kind of predict future experience with the products that are in development and the products that are coming in. So if we can actually identify the risk factors or the characteristics of the products that worry us and the products that maybe we don't need to be so worried about, I think maybe we can make some progress that way. That's the hope, and that's what we'll be trying to cover in our regulators only sessions as well. Anyone else?
Thank you. Go ahead. Dr. Marie, please.
I think the fact that we build this the guidance on our experience is entirely logical, though, but it doesn't say that for future molecules it will be possible. What we are saying at the moment, based on in-depth knowledge of a reference product, but as they are at the moment, those we have seen, it is possible to predict the efficacy based on the mechanism of action and then to study all the biological functions which are relevant. But it's possible that some new molecules, we will not be able to do that. And in that case, we will have to do some efficacy trials. So and the notion of risk, I'm not sure I understand. Is it an analytical risk for some type of molecules? I don't know, maybe bispecific antibodies with payload, those things which are becoming very complicated. It may not be possible to analytically study them enough. And I'm being the clinical assessor, I have no idea. So is that this kind of idea you have based on the structure of the molecules? But I wouldn't think that it can be based on risk, clinical risk, saying, oh, this is for oncology, so we need to be much more careful. This is just for headaches, so we don't need to be, it's not as important. And so I don't, but I don't think that that's what you have in mind. I'm just saying that the notion of risk needs to be, yes, to be discussed. But I think for the future, it's very difficult to predict what we will see. So we can only say, based monoclonal antibodies now and fusion protein, it's something that we know all the mechanism of action, CDC, ADC, apoptosis, blah, blah. So it's well known, and that's why we can, and we have functional assays for all these. So we are starting to be really comfortable with this. But for things completely different, I agree, we don't know. So I wouldn't say that the future is absolutely without any clinical trial. I had a question about the quality of the data we see now, and I don't know whether other regulators have the same impression, is because companies are, because the molecule are known, especially if it's known molecules, and they make less and less efforts, and because, oh, this is known, it doesn't have any impact, so we don't need to do that. And that's a trend which worries me, because I don't think that it should go in the direction of less in-depth analytical analysis rather than more. And there was this comment also, whether should it be down to the company to take the risk of not doing a clinical trial. And yes, for me, yes, they are the people, they know their product, they should know the reference product really by heart, and they know the target and what they want to achieve. So yes, it's their risk. They should come with, as we have repeated now, with the demonstration that they think that most clinical characteristic can be predicted from the analytical. And no, it's not down to the regulator to take the responsibility and say, oh, this is a known molecule, so you don't need to do a clinical trial, or you need to do a clinical trial. And I think we have really to emphasize that the manufacturers are the people who are responsible and should show that their product can be approved, maybe without any clinical trial. I'm stopping now.
Thank you, Marie-Christine. What does the panelists think in regards of that the manufacturer should be capable or able of answering what is or not what is not required? Looking forward to the manufacturer session, and I want to see what they have and what is their experience in dealing with regulatory authorities, especially to this regard. I want to ask Hina, Dr. Hina, did you have an experience where the manufacturer wasn't really sure or clear on what was required by the WHO, or what are the scientific requirements for getting their products approved? What are your thoughts on that, please?
Yeah, actually, we don't have a direct connection with manufacturer regarding the issues, but maybe I can answer to your previous question regarding the harmonization of regulatory framework. These days, we don't talk about a lot of regulatory harmonization, but talking about the regulatory convergence, because now we realize it's like an impossible mission to change the world and to get the harmonize every regulatory framework, not only for biosimilar, but also for all biologicals. But what we understand is true, it's impossible to quantify the residual uncertainties. But what we can see from the a survey or a review conducted in 2019 and 2022, we understood the degree of residual uncertainty is not as high we experienced in 2009. That means the experience we have gotten from the development and evaluation of biosimilar affects to the flexibility and also to increase the confidence and familiar with the biosimilars. So it's difficult to predict the future, because we don't know what we would experience in the future, but the experience would affect to the change of regulatory framework in the future. Over.
Thank you, Dr. Rina. We have a question in regards of post-marketing surveillance and how it can be utilized going forward in biosimilar development. How can it be more utilized? Dr. Marie-Christine, please go ahead.
Our position is that a biosimilar should be demonstrated to be biosimilar at the time of the approval, and that no data afterwards can be requested to confirm or change that. If we are not convinced that it's a biosimilar, then we should not approve it. But the post-approval data are, we don't think that they are absolutely critical, except when there are some specific issues with a different device or a different formulation where it might be important to get more data. But in principle, certainly not about efficacy. We would not expect more efficacy coming post-approval. And safety, as we think that the safety of the reference product is, the safety database of the reference product is the basis for the safety of the biosimilar, we don't think that anything much can be added. So except in very specific cases of differences, for example, as I said, device formulation, we don't expect anything from post-approval studies, and we need to be convinced at the time of the approval that it's a biosimilar. If not, no approval. Thank you.
I'll comment on that as well. In Canada, we do require pharmacovigilance and standard risk management plan, as we do for all drugs, but completely agree that the biosimilar has to be proven biosimilar to the reference product. It just simply doesn't make sense to put a copy of a product onto the market when a proven safe and effective version of that product is already on the market, unless we are convinced that this is as safe and as effective. But that said, we do have pharmacovigilance activities in place for all drugs that we authorize.
And I can add to that as well. There is nothing different. We are certainly agree that if it needs to be demonstrated to be biosimilar. So in terms of post-approval changes, I can comment from the pure manufacturing perspective. We would follow ICH Q5E. And it's impinging on the company to demonstrate that following a change, the product is comparable to the pre-change product.
Maybe also to briefly add up from the European side, this would not be a viable option for us to determine on comparable efficacy post-marketing. We have a regulatory, technically, as soon as an authorization is granted, the product stops being a biosimilar. It is treated like any other product. So no further liaison to the originator can be expected. And I also think it wouldn't make sense, because in biologics, we know that with manufacturing changes, etc., within certain ranges, but they might shift a little. And that should all be clarified at the time of marketing authorization and cannot be accounted for post-marketing.
Thank you, René. We have a question for you in regards to future direction of guidelines? And is it going to be product specific or more of a generalized guideline for biosimilars?
We're having that discussion. I mean, overarching concept paper followed by a reflection paper with a bit more detailed principles. We have no specific plans for product-specific guidelines that might come. However, I have to say, I think it's quite a challenging exercise because which antibodies would you focus on? There are so many, and it is maybe not realistic to have a product-specific guideline on every one of them. And again, being realistic, also the knowledge, as soon as we have the knowledge for a specific monoclonal, it might not be an interesting target for biosimilar developers anymore. So there are still a few things to be considered before that decision is made.
Will it be the same for the other regulatory authorities as well, or is there a different approach that be implemented?
In Canada, we've generally stuck to a single guidance document that is very general, and we'd encourage sponsors to discuss with us their plans for specific products.
And we don't have product-specific guidance at this time. Obviously, we also recommend sponsors to come and talk to us on a product-specific basis.
There's one question I see in the chat about the consideration of having a small clinical study done in lieu of, say, a statistical evaluation of PD or efficacy to, somehow fill the gap. And it's an interesting concept, and just, would those data, a descriptive analysis of patients comparing endpoints, I guess, specific to whatever the patient population is and in their response to the treatment, give any greater confidence in the utility, give greater confidence for, really the healthcare prescribers. I think at that point, from an agency perspective, I don't know how convincing the data would be, given our penchant for hypothesis testing and statistical evaluation. So is this something that other regulatory agencies have ever considered? I've heard it called a comfort blanket to have these clinical efficacy studies. Would a small descriptive study provide that same kind of comfort?
I don't think that's something that we've really considered at Health Canada, but it's interesting that you bring up the term comfort blanket there, and we've been part of a lot of panels and for Canadian health professionals and patient groups, and there is a level of comfort that comes to them and helps with uptake when clinical data is available for them to compare to their reference product, especially a lot of products that where patients are being required to switch from a reference product to a biosimilar. But as a point of demonstrating biosimilarity, I'm not sure what the value is from that point of view, but certainly to promote uptake, there may be.
My problem with descriptive trials is they're great when they sort of give you results that you are expecting, but they're terrible if you're trying to evaluate numerical imbalances. There was a question in the chat earlier sort of about differences in adverse event rates. And, you know, this is always a difficult thing, right? Because a lot of that can be stochastic trials, even with, you know, bioequivalent efficacy endpoints don't have that for safety. So there's a lot of information in a trial that's descriptive and can just by chance have an imbalance. So, you know, doing a small descriptive trial is maybe good, but to me it's kind of risky just if we're just doing it for comfort.
Thank you, Sarah. We have a question about Biosimilars. It's asking if it needs all the requirements for biological registration or maybe more close to biosimilar registration requirements.
That's a tough one because I think all the different countries may have different sort of legal frameworks for evaluating a biobetter. In the US, a biobetter has to follow the 351A pathway right now, which is our, you know, primary biologics approval pathway. So they would fall out of the biosimilar pathway if they were going to differ from the reference product.
Same in Europe, there are no biobetters. They would be standalone biologics. That doesn't exist here.
We in Canada would consider that a new product entirely.
Dr. Marie-Christine, please.
Yes, I don't know what you call a biobetter, but we tend to have more and more of these cases where the immunogenicity is more favorable because of either another cell lines or less impurities or improvement in the manufacturing. Again, I'm not the expert for manufacturing, but as far as I remember, the EMEA guidance, and we are still very much following that, we would not preclude and refuse biosimilarity to a product which would be less immunogenic. As long as there are no claims of being better regarding better safety or better efficacy, etc., we would accept it as a biosimilar, because if not, we would really penalize better products and ask them to do a full development, while as a biosimilar, they can have 10 indications. So there is this question, but maybe that's not what you had in mind when you are talking about biobetter. It's when you really have a target to do a better product in terms of better efficacy or better safety. But we have more and more of these cases where we have less immunogenic product. I don't know what Rene thinks about it, because he probably sees them, and we had the discussion a long time ago at the EMEA about what we are doing about these products.
You're right. I mean, the consensus is that we accept the better immunogenicity also in order not to shut down a good product. However, it would become a bit, I mean, for most cases, we still have the efficacy trials, and it would get complicated, I would anticipate, if the immunogenicity would be really a lot better, and then the trial also overshoots to the upside. That would be perhaps a real problem. If it doesn't show in the efficacy trial regarding the primary efficacy endpoint, I think it will always be accepted.
I think my understanding for BioBetters is that there's purposeful modification of the molecule in order to improve its performance, in which case I think if you've purposefully modified the molecule, then it won't be a biosimilar product. And that is, so we don't have any separate pathway. We actually don't have a separate pathway for biosimilars. They're reviewed as new drug submissions in Health Canada, and that would be the same for BioBetters, but I don't believe that we would follow the biosimilar pathway in terms of development.
We have a question about reference products and if the US FDA would be willing to accept the reference product in Europe and then vice versa. So what's the regulation in that regard, and are there updates on what kind of, or which reference product can be or cannot be used between different regulatory jurisdictions?
I mean, acknowledging that this is like a sort of a factor that maybe prevents optimal streamlining in various programs. I think it's kind of an unresolved policy issue for us, whether we can and how we can sort of utilize reference products from other areas. I mean, at the moment we do scientific bridging, and what that entails can differ. But it's, you know, our laws sort of requires a comparison to the US reference product. So we sort of have to figure out how can we fulfill the law and yet allow use of you know, reference products from other areas. It's an area that we discuss not infrequently internally. But unfortunately, I don't have a set answer for everyone today.
And then the follow-up questions that would be a participant kindly asked if the FDA and EMA agencies or any other, all the agencies would be open to have a joint scientific advice meeting on tailored development concepts. I know there are several cooperation programs by different regulatory authorities, but I'm not really familiar if there are any advice meetings that are carried out between different regulatory authorities.
Stacy, do you want to talk about the parallel scientific advice pathway, maybe, or?
Sure. There is an opportunity for a developer to request both the EMA and FDA discuss the questions regarding their proposed program, and we would the two agencies meet and consider the questions and provide responses in a collaborative manner. So there's information available on the FDA website, I'm sure on the EMA website as well, with the details about how to go about requesting those meetings.
And is that specific for biosimilar products, Dr. Ritchie, or for any type of product?
I believe it covers different, new drug development as well. Yeah. We can look for the link. Maybe share it. Absolutely.
Thank you. Okay, we only have a few minutes remaining. I want to open the floor to the panelists to have any final thoughts or remarks. So please, anyone, go ahead.
Well, I want to thank everyone for participating today. It's obvious there's a lot of interest in what we have to say. There are a lot of questions that we didn't get to. Some of them maybe a little bit too program specific or product specific for us to really tackle today. We will try to include a list of questions with the workshop summary so everyone can see the range of them. And I think what I would suggest as a sort of take home for folks is that I think there's a real spirit of collaboration and cooperation, and we do want to try and be consistent across agencies. I think we all have different sort of frameworks to implement or slightly different frameworks to implement. And then we also have sort of the challenges that just go along with trying to get a big group of humans to think the same way about something, right? I mean, I think there's just, I think we who are very familiar with biosimilars are pretty comfortable with the idea of streamlining. I do think that there is some, there are some other factors that sort of go into play in terms of what the various folks end up, where they end up falling in specific recommendations. So just to say, I think that we're making progress. And I think just even in the last few years, I've seen some shifting in people's comfort levels. So I think there's hope. I think that we need to follow the science and what the science tells us. And so that's our goal anyway, to sort of head that way. But we really appreciate all the communication and efforts with our colleagues around the world. So so appreciate your participation.
Thank you, Sarah. I would like to thank the panelists and I'd like to thank the participants. I want to give it to Carol to give the final remarks. Please, Carol, go ahead. Thank you.
Yeah, thank you, Dr. Ali Omidin, for what an excellent moderation, and thank you all speakers and attendees, providing a really good question, and the questions are just keep flowing in, and we're just gonna have to go through them after this meeting, and we hope to address them with the regulators and have further discussion. So hopefully that we will be able to have a nice summary of the outcome of those questions and answer that will be available to the public in future. And I want to thank all the speakers and attendees, as well as the panelists, all of you, because if it weren't for you guys' help and dedication, this workshop wouldn't be successful. So thank you so much for doing that. And I do want to apologize for some of the technical gaps or difficulties that we had a few moments here and there, but I want to say, thank you so much for the AV support. Marcus, you've been great, and it's been really a pleasure working with you. And I also want to mention that 3 hours is a very long hours, just sitting without a break. So we are thinking about having maybe a short break for tomorrow's session so that we can have a little bit more, a break rather than just continuing for 3 hours. So just to remind everyone, tomorrow we will have a open public session, hearing from the respective stakeholders perspective on what they think about having the comparative efficacy studies in a biosimilar development program. And thank you all for your time. And I think we can adjourn now unless anybody has any questions.
Thanks, everyone. We'll see a bunch of you tomorrow.
Thank you.